talked about both DNA and RNA as being these linear polymers. And of course, they're made of these things called nucleotides. The nucleotides themselves have this ribose backbone, this 5-carbon backbone with a nitrogenous base, hydrogen, hydroxide, and then 3 phosphate groups on the 5' carbon. So we can call these the 1' carbon, 2' carbon, 3', 4', 5' carbon. And we call these nucleotide triphosphates. And of course, these are what are being used to make that backbone with a phosphate backbone. We dealt with the different types of bases, of course, purines and pyrimidines. So remember, for your purines, these are basically your adenine, guanine, and remember they're composed of 2 cyclic nitrogen rings compared to the purines, which are just a single nitrogen ring, or excuse me, pyrimidines. So your pyrimidines are cytosine, thymine, uracil. So when we describe, I think I talked about this before, when we describe the bases by themselves, we use this terminology. When they're actually part of the nucleotide, remember, we get a name change. And sometimes I mess up and I use them back and forth synonymously, but usually I try not to. So once they become nucleotides, so nucleotide trisephosphates, we would have adenosine instead of adenine, guanosine, cytidine, thymidine, and uridine. But most of the time when we're talking about the sequences, these A's, T's, C's, and G's, we refer to the base name. So don't get confused. We're basically using them synonymously most of the time. It's just a difference whether it's the base by itself or a base that's attached to the ribose backbone that we talked about. We also described this linkage between the monomers and how that gives us directionality. And so for any given nucleotide, here we're just going to make DNA. It's going to have a before I give it to you. That's right. And anytime I give you the PI, that's just a shorthand for phosphate. So we have 3 phosphates on that CH2, 5' CH2. We have this phosphate bond that's created between the two. And we kind of start to develop these linear forms of DNA and RNA. And remember, we kind of do this in a 5' to 3' direction. Remember, 5' is just the 5 carbon, 3' is just the 3 carbon, and all this means is that we keep putting new monomers up to the bottom and connect them to the 3'. So we're basically just adding units from the 5' to the 3' down. We do this for both DNA and RNA. But it also gives us our directionality. Being white male in science, we tend to write things very two-dimensional in cartoonish, Australopithecine form, if you will. So we describe this directionality usually as just a line with a 3' side and a 5' side. So that 3' always has this free hydroxy, so we can always kind of put that on there just so you see there. And this 5' carbon is always going to have this 3' or 5' phosphate. So we can just kind of put that there as a CH2, 3PI. And it gives us this directionality. It's kind of like the directionality of a protein. One end looks one way, one end looks the other way. So we get these nice linear strands of DNA. And of course, when we look at the strand of DNA, we have the ribose backbone kind of pointing out, and those bases usually pointed to the inside. Now, all of the structures that we talk about here, by all those different types of bonding forces, the van der Waals we talked about, electromagnetic, hydrogen bonding, etc. So we get this nice linear strand of DNA. Now DNA rarely, unless you're a virus, hangs out as single-stranded. It's very reactive, and it quickly wants to bind up with something that has a homologous sequence. So when we talk about our double-stranded DNA helix, once again, we think of this as an inverse complementary arrangement between two strands of DNA. So inverse complement, so that means when we draw our strands of DNA, on one strand we're going to have the 5' side. Because it's the inverse complement, we're going to have the other strand as the 3' side, and vice versa. And we have our A's and T's, hydrogen bonding with our C's, or excuse me, hydrogen bonding with our T's, and our C's hydrogen bonding with our G's. It's been a long day already. So it's actually the CGs that give us our melting temperature. We call this the G to C ratio. And what that means is when we get above that melting temperature, these strands denature into their single strands until that temperature goes down, and then they zip right back up. So this melting temperature is based upon the hydrogen bonding that we see. But this double-stranded DNA, as we said, can be manipulated with not just heat, but other things as well. So denaturing double-stranded DNA basically makes it into its single strands. We talked about heat. Heat will denature it. And we think of, for mesophiles, those are us. Those are organisms that like temperatures between, 15 degrees and 37 degrees C. These guys have denaturing right around 85 to 95 degrees C. And those strands just pull apart. You drop the temperature, and surprisingly, those strands will just anneal right back up. So we also can do it with increase in ion concentration. So think of this as increasing the types of salts you put in there. Those salts compete for the hydrogen bonding, also rip it open. Then things like acids and bases, mild acids and bases, can all be used to denature these strands of double helix DNA, if you will. So when we talk about a sequence, and many times you'll just get these sequences thrown out at you, specifically if you're on the internet looking stuff up. Usually describe it from the 5' side to the 3' side. So if I'm going to give you a bunch of A's, T's, C's, and G's, as I'm giving it to you, I'm not going to tell you it's the 5' side or 3' side. We just usually assume left to right that we're reading in a 5' to 3' direction. It's just this commonality throughout all DNA sequencing. Now, RNA can be pretty long, and DNA can be pretty long, much longer than the polymers we've been talking about. And we describe the length of these polymers based upon what we call either nucleotides, if it's a single strand, or base pairs. So if it's single-stranded, or single-stranded RNA or DNA, we can talk about it in nucleotides long. If we're talking about double-stranded DNA, we describe this length in base pairs. Basically the same thing. So for most organisms, we have chromosomes that are pretty long, unless you're a prokaryote. So we usually describe them in either base pairs, 500 base pairs of DNA, or kilobases, 500 kilobase pairs of DNA. And recognize that these polymers can be really long. Millions and millions of base pairs long in one strand. Pretty incredible. So in your genome, if we take a look at any one of your cells, the genes in your cells, you have about 3 billion nucleotides. It's a lot of DNA. So we're always trying to condense these things up, and we'll talk about that as well. So we kind of have this double-stranded DNA helix. And we talked about some of the intrinsic secondary structures. That it's a right-handed helix, meaning it turns right-handed. It has this kind of central axis. Has both major and minor grooves. And like we described before, this intrinsic structure, due to the hydrophobicity of those nitrogenous bases, those bases get tucked into the inside of the helix, and we have those outer ribose backbones kind of on the outside. Recognize that due to this length of DNA, I'm not sure if we covered this before, it's got to be condensed down to fit into the cell. So for prokaryotes, remember this is a single circular chromosome. Here's my double-stranded helix. We basically do what's called supercoiling. And just think of it as taking a rubber band and you start to twist that rubber band up. It starts to fold upon itself and starts to form these big globular shapes. And that's what we can see with this as well. as we start to wind it up, it starts to supercoil. And we do this so that we can condense it down enough to pack it into the cell. With eukaryotes, we form chromosomes. So with eukaryotes, we condense them with this stuff called chromatin protein. Specifically, these things called histone proteins. And so if we take a look at your chromosomes, they have these big millions of base pairs of linear DNA. So these are circular. They don't have free ends. These are linear. They do have free ends. So what we end up doing is spooling the DNA around these histones. Here's our double-stranded DNA. And there's a series of histone proteins, 5 to 6, that usually hang out. And they're kind of like the spool that the DNA winds around. So these are our histone chromatin proteins, excuse me. So anytime you hear the term chromatin, that's just a way that the eukaryotes condense those down. And as we know, spooling is one of the most methodical and easiest ways to take really long linear strands and condense those down. I think we talked about the different types of DNA helices, the B helices, the Z helices, the A helices, I think, didn't we? So remember that there are different types of helices. B DNA is basically physiologically active and normal. We have A DNA, which is basically dehydrated, if you will. Inactive. We have that weird stuff called Z DNA. We're really not sure what it's for, but it has all these GC repeats. And we've actually, I don't think to date, isolated a Z DNA helices outside the lab in nature. But we know they exist because there are viral proteins that are made specifically for Z DNA, so they may have something going there. And then, let's see. I think we talked about the beginning of replication. So we'll deal with DNA replication first, and then we'll come back and talk about the differences of DNA and RNA. So DNA replication, as we've talked about, it's also directional. It also happens in this 5' to 3' direction. It's carried out by this really cool enzyme called the DNA polymerase. So basically this replicates and proofreads. So it has the ability to back up a little bit, recognize an error it's made, fix that error, and then continue on. And it happens in a semi-conservative nature. And by that we mean the template or parent strand becomes part of the new DNA duplex. So what we find is that these template strands, here's our template, they denature into their single strands as templates or parent strands. And then during replication, those parent strands now become part of the new DNA duplex. So the blue ones are the new strands. And this is why we call it semi-conservative. If it were conservative replications, the new DNA strands would only associate with themselves, and the template strands would only associate with themselves. They wouldn't basically make this kind of mixture of a duplex, if you will. And then, of course, we showed you the replication fork, and I think that's where we stopped last time. Before we go there, let's talk about this polymerase a little bit. So this DNA polymerase that's making these new DNA strands, basically that 5' 3' phosphate bond between the nucleotides, as far as its proofreading goes, it's fixing mistakes that are made during replication, that the actual enzyme it made itself. So for instance, let's say that we have an A that should be bonded with a T. But during this process, we get a transversion. And A is bonding with a C. The polymerase just made a mistake. It put the wrong nucleotide in when it should have put in a T. And like I said, it has this ability to kind of back up and go the opposite direction slightly, recognize what that is, clip it out, and insert the correct nucleotide. So this is different than fixing mutations. Basically, when we fix mutations, these are different systems called mutation repair systems. And we're not going to get into those. Basically, it's a semester in itself talking about those.
Is there like a short way to describe how it fixes them or knows how to fix them?
It isn't that complicated. I guess the best way to describe it is the activity of the enzyme. And I'll just do it really quickly. So this DNA polymerase has various domains, like these proteins we're talking about, right? The various protein domains have various functions. So one of the protein domains is all about facilitating that phosphate bond and replication. And so what it's doing, its main action is to grab new nucleotides, put them together, and create this phosphate bond between the nucleotides. The second domain, the other side or part of the protein, is basically what we call an endonuclease. So endonucleases are basically molecular scissors or shears. And so it has the ability to cut out the bad nucleotide and replace it. It's doing this based on the template. So it has the ability to keep reading the template, and even though it recognized it made its mistake, based upon the template, it can still recognize what should be there. If it goes too far forward, it can't go back far enough, and then it's just there, and a mistake is made. Now, a lot of the questions that are asked are, how do you know which side's the template? Right? Template strands are differentiated from new strands because they're modified a little bit, what they methyl group. We end up putting a whole bunch of CH3s on the template strand so we can always delineate which is template and which is not. And that's for the enzyme's sake. The enzyme can recognize that. So I don't know if that answers your question or not. And so, of course, we talked about the replication fork. And we're not going to go into it again. We kind of talked about it, right? And recognize that it starts, once again, at this origin of replication, this ORI we talked about. We're not going to go through the whole process. We're just going to talk about some of the generalities, the attributes to that replication fork. So if you're a eukaryote, we have many origins of replication. And each one of your chromosomes, your 23 pairs of chromosomes, 22 pairs of chromosomes, one pair of sex chromosomes, which aren't pairs. There are basically ories located all throughout those chromosomes. And it allows for these things to be replicated in many different ways. But during this process, it's unidirectional. As this fork opens up, it just basically goes in one direction. With prokaryotes, it's a little bit different because they have just one single circular chromosome. They usually have one origin of replication. And they basically have this multi-directional replication fork. So in this case, if I were just to show a eukaryote, here's my origin of replication. I've unwound it and getting ready to replicate it. That polymerase just goes in one direction, 5' to 3'. With prokaryotes, they're circular. So it's more like putting a inner tube inside of a tire. Because we have this bidirectional, here's our prokaryote. It's bidirectional. So basically we're going in two different directions at one time. And it just does the whole chromosome in one go. So there are these slight differences, but not too much. So just kind of DNA replication in a nutshell. Not going to go too heavily into it. Now, I don't know if we talked about RNA molecules or not, did we? We'll just kind of quickly gloss into RNA. So for RNA, the only differences that we see is that we use ribonucleotides rather than deoxyribonucleotides, and the only difference is on that 2'. On that 2' hydroxy, we have an extra oxygen group here. So for DNA, this is only a hydrogen. For RNA, it's a hydroxy. It's an OH group. That's the only difference between the actual ribose backbone. Then for our bases, of course, we use A, G, C, and uracil replaces thymine. So if we take a look at an RNA molecule, it might be AUG, CCG, whatever. And we can see that that U is replacing the T's. Same thing, it replicates and is directional as well. It has this 5' to 3' directionality. Replication also, just like DNA, also happens in that 5' to 3' directionality. Just like we saw, we get that nice phosphate bond between ribonucleotides until we get a nice long chain of RNA. Now, RNA is very rarely double-stranded, but usually RNA is single-stranded. It's linear, but folds on itself to form these secondary structures. And we've talked about all sorts of messenger RNA, but there are all types of RNA out there. So messenger RNA, of course, encodes proteins. tRNA is a transfer RNA that brings us amino acids during translation. We have all sorts of regulatory RNAs like silencing small interference or microRNAs. And so these are silencing microRNAs. And these are all about regulation. They have the ability to turn genes on and off. We have these things called small nuclear RNAs. Also help in expression. So there are a bunch of different types of RNA molecules out there. Now I'm going to use tRNA as an example of this secondary structure, and because we're going to be kind of glossing over expression here in a few moments. For our transfer RNAs, they have a specific shape. As such, 2' man, 5' man, they have all this secondary shape of bonding between the two. On one end, we have our amino acid. And on the other end, we have the anticodon. And so basically, these are used as a transport molecule just to bring amino acids in when we're making proteins, during protein synthesis. Really nice secondary structures, has its own shape and function once again. Even though we see it much more on the protein level, we still see it also a little bit on the nucleic acid level as well. I have that backwards. My bad. Amino acid goes on the 2' end. So I'm just going to give you a quick overview of expression. I'm sure we've all seen it millions of times, and we don't need to go through it too much more. But we'll bust upon it a little bit. So the two main things we see, this replication, and then the second thing we see is how we express our DNA, and it's a much more complex process. Like that dogma we keep talking about, DNA is transcribed into messenger RNA and translated into protein. So we'll start with the transcription process. Once again, we're not replicating DNA, we're actually starting to read the sequence now. So let me back up even a little bit so it makes a little bit more sense. Genes in DNA are organized into what we call an operon sort of organization. We first found it in bacteriophage viruses, later in prokaryotes, and of course, this goes all the way up to eukaryotes as well. So I'm no longer going to just draw my double-stranded chunks of DNA, but going back to kind of a linear block-like representation of what's going on. So here we have what we call an operand. What we would have upstream is something we would call a promoter or a regulatory sequence. And the most common one is called the TATA box, T-A-T-T-A. That TATA box recruits the transcriptional machinery. Downstream of this, we have our actual genes. I'm just going to call these gene A, gene B, gene C. These, when translated, will give us protein A, protein B, and protein C. So these are the encoding sequences. Almost all of our gene organization is organized like this. We can have other regulatory things upstream as well that are like enhancers. But for the most part, the promoter and those genes are what we need. So this is the DNA that we're looking at here. What happens is, during transcription, you get an RNA polymerase recognizes the promoter. That, and along with a bunch of other transcriptional factors, we call TFs. And there are various families of TFs, 20-something different proteins act as transcriptional factors. They all come together on the DNA. The RNA kind of slowly unwinds it, reads the template strand. RNA polymerase basically forms the messenger RNA, once again in a 5' to 3' direction. And as we said, U's are basically replacing T's. So that's the beginning. All of a sudden we get these translational factors show up. They actually dimerize on the promoter, and they start transcription. So we're going to end up with this, with an A mRNA. You're going to have a B mRNA, and you're going to have a C mRNA. Everyone okay with that so far? So we're just transcribing the the DNA into RNA. The second thing that takes place, of course, is that with prokaryotes, mRNA goes straight to translation. And with eukaryotes, we get mRNA modification. In eukaryotes, we don't like that native mRNA to be used. We like to modify it a little bit. Eukaryote is made of two different types of DNA. Exon and intron DNA. We don't see that in prokaryotes. Well, I used to say we don't see that in prokaryotes until someone looked it up and found that there are those exceptions where some bacteria do have intronic and exonic DNA. And so one of the things we do during eukaryotes is we clip out all the introns. And we recombine only the exonic mRNA. So we clip out the introns. We give it what's called a 5' methyl cap, guanosine methyl cap to be specific, and a 3' polyadenosine tail. Then it goes on to translation. So there's a little bit of modification first before eukaryotes go ahead and transcribe or translate theirs. So we have prokaryotes that transcribed their mRNA. Now the next step is translation. Translation starts, of course, with the mRNA. A few main players. The ribosome complex, which is the big one. Transfer RNAs. And there are tRNAs, a different one for each amino acid. So there are 20 different transfer RNAs, each one specifically for their amino acid. And dealing with our mRNA, and we'll talk about this. Translation starts at the first methionine codon. So if this is my mRNA here, and basically what I would have is an AUG, which is a methionine codon. I'll just call it MET so you don't get it mixed up with methyl group. And we start to read the mRNA. In triplet codons. So 3 base pairs at a time. So the next one would be CCG, GUG, so on and so forth. And so we could put in our leucine, valine, etc., etc. These would be our amino acids making up our peptides or proteins. So, as this goes, we need to describe it a little bit better. So it's read in triplet code, these codons, as we just talked about. 3 base pairs at a time. And for any given amino acid, we have a degenerate code. Degenerate. Anytime you hear that term in science, doesn't mean you're hanging out on the corner smoking pot.